Welcome to this week's episode of Brainstorm, where we give you a glimpse into the world of science for Wednesday, June 5th, 2013. We begin with news from the world of chemistry. If you are a longtime viewer of Brainstorm, you know we sometimes cover developments and technologies that could help mitigate climate change and greenhouse gas emissions. Usually these stories only address part of the problem, and solving this issue entirely will take multiple efforts. But sometimes we get lucky and can cover stuff like a recent development from the Lawrence Livermore National Laboratory. As a civilization, three of the many things we need are clean and renewable fuel, a way to counteract CO2 emissions, and a way to protect our oceans. All of these climate change-related issues may, and we stress may, be solved by a single process. The Livermore scientists demonstrated that electrolysis, the splitting of water with salt water and added silicates, effectively produces hydrogen. The waste solution from this chemical reaction is highly alkaline and actually effective at absorbing CO2 that is bubbled through it. After which, that still alkaline basic solution could be added to the ocean to counteract ocean acidification. For all three uses, hydrogen production, CO2 sequestration, and ocean deacidification, this chemical process may be cheaper, but more study is needed. Still, it does use cheap and abundant materials, saves time and money by using the byproduct directly instead of recycling it, and the whole process can be even greener if the electrolysis is powered from renewable sources like solar or wind. It'll obviously take time and development to scale up, but it is still exciting news from the world of environmental technologies. Next is a story from the world of biotechnology. Ultrasonic sound waves have found applications in medicine, such as ultrasound imaging and breaking up kidney stones. Now, researchers at Iowa State University think ultrasonics may be the key to economically viable biofuel production. They found that ultrasound can be a more effective and potentially more energy-efficient way of processing biomass at certain stages. Potential feedstock like corn stovers, switchgrass, and softwood could be pre-treated with ultrasonics rather than using enzymes. This step is essential in biofuel production as it removes the structural protein lignin from the cellulose sugar, which is the target. Another application is the breakdown of cornstarch into glucose, which is then fermented into ethanol. Normally, corn is turned into a starch mixture using high-temperature steam to break it down. But ultrasound alone can smash the corn into small particles, providing more surface area for the enzymes involved in the next stage of processing. This provides comparable ethanol yields to steam treating, while potentially using less power. A further application is the processing of vegetable oils like soy into biodiesel, with the primary chemical reaction, called transesterification, accelerated to less than a minute with the addition of ultrasound. In addition, yeast starved with glycerin produces high yields of an oil that can be simultaneously converted into biodiesel with ultrasonics. All these applications increase the economic and energy potential of biofuel technology and will hopefully lead to an increased use and development. We end with news from the world of medicine. In the past, we have ragged on us humans' poor regenerative abilities, but we aren't completely helpless. Arguably, the organ in the human best at regeneration is the liver. Parts of it can grow back if damaged or removed. Still, severe disease of the liver often requires a transplant, and coaxing liver cells to grow outside the body has had little success. Those challenges may be a thing of the past, however. A group at MIT working with other institutions has discovered a variety of compounds that encourage normal liver cell functioning outside the body. This was possible because they had previously developed a way to temporarily stabilize human liver cells called hepatocytes. The trick was carefully intermingling them with mouse fibroblasts. Those are the cells that create the extracellular matrix, the mixture of proteins that provides structure to animal tissues. By creating many small samples of these stabilized hepatocytes, they were able to screen 12,500 chemicals to see how they affected cell functioning. In the end, 12 compounds were shown to either stabilize the hepatocytes or even encourage further cell division and new tissue growth. Also very encouraging, these compounds helped fully mature induced pluripotent stem cells into functioning liver cells, which has previously been a challenge. Other researchers have begun testing the successful chemicals with other cell types to see if they are a universal trigger of cell maturity. And the original group will continue working with human liver cells, these new chemicals, and cellular scaffolding in the hopes of creating artificial liver tissue for transplanting and saving lives. Well, hope you enjoyed this episode. In reference to our first story, what multi-purpose invention to help the environment would you create? Let us know your thoughts on that and all the stories in the comments.